I'm Dr. Lisa Schwanz. I'm a senior lecturer at the University of New South Wales in the School of Biological, Earth and Environmental Sciences. I do research on the evolutionary ecology in animals, and I mostly focus on how animals respond to change in their environment or change in their own physiology. I'm also an educator here, so I teach undergraduates and I supervise honors and PhD students as well. Well, I always loved spending time in nature and with animals. I've just always been interested in animals. I suppose when I was in high school, I learned about the research of some famous biologists, such as Dian Fossey, working with mountain gorillas. And I just found her work on conservation of animals and her science and just the devotion that she showed to working with mountain gorillas really inspiring. And so I decided to pursue working with animals and conducting science on them. And then when I went into my education at university and throughout my PhD, became really excited in the science of understanding animals in their environment. So right now I'm working with a small lizard that lives in Australia called the Jackie dragon. And I've been quite interested in how the development of animals into males or females can depend on their environment. So in a lot of turtles and in a lot of Australian lizards, whether they develop as a male or a female depends on the temperature of their egg when they're developing. So I wanted to understand that, both the evolution of it, why does that exist, but I also wanted to know what it means under climate change. If the climate gets really warm, does this mean that the sex ratios will become very biased and imperil populations? So I decided to focus on a species of Australian lizard that I could study experimentally. So I wanted a small animal that I could bring into captivity and do focused experiments on it for some of the questions I wanted to ask. I suppose a lot of it is just kind of mind mapping ideas and trying to figure out what's interesting, what isn't known in the field yet, what needs to be known to advance the field. And then when you're thinking of those questions, trying to figure out the data that are available or the animals that exist or the places that exist where you can go and find answers to the questions that you're interested in, and then developing your hypotheses into predictions based on those. So I've worked before with a turtle in North America, and that turtle, there was a big data set of them wild, just living in the Mississippi River. And it was fantastic data from natural nests looking at sex ratios and climate. And I found in those turtles that it looked like in the warmer years, there was something happening that looked like the parents might be influencing the way that the sex ratios were developing. So I wondered if it could be related to hormones in the yolk, which we know can affect sex ratios in reptiles as well. And I wanted to do a controlled experiment to specifically ask that, because with the wild turtles, we only had what nature was giving us, and we could only look at correlations. And I wanted to look at causation, so I wanted to design an experiment where I could manipulate the temperatures that parents experienced and see if that would influence the sex ratio that developed, not just the temperature that their egg was at, but the temperature that the parent was at. So for the big reptile development database, I've worked with some researchers here in Australia, so with colleagues at University of New South Wales and University of Tasmania, and then as well with collaborators and colleagues from Lund University in Sweden, from Auburn University in the US, and from the Chinese Academy of Sciences in China. And these were all researchers that were interested in the question and had been putting work into it. So we all got together, combined our efforts to put this big database together. So to some extent, they can be people you know personally. So some of the people I have either worked with in the past or I work with currently. They're people that I see at conferences, just colleagues that I know, their research. And you just sit around and you talk about the science you're doing. And this database that I'd been working on for a couple of years with someone here at UNSW, we'd been just talking to some other researchers, and they said, oh, we tried to do something like that 3 years ago and we just couldn't, it was too much work. Do you guys want help finishing it off? Could we all collaborate together? And we said, that sounds fantastic. So we all just combined efforts so that we could I think, improve the science with a larger number of people and more and different perspectives on the data. We're all reptile biologists to some extent, but we have different foci on what we do and what we're interested in, just united by wanting these data to ask different questions. So some of the researchers are very interested in the actual developmental biology of reptiles. Some are interested in what it means for invasion biology. Some are interested in what it means for response to climate change. Working with vertebrates has a lot of ethical guidelines that you have to follow. So the first thing you have to do is consider the ethics of research. You have to get ethical permission from your university to work with them, and you have to get permission from the state to hold them and to collect them. So those are some of the first planning issues. You also have to get money to feed them and to keep them going, because they're quite a lot of work to maintain. So those are some of the logistics. Well, one of the tricky parts, I think, about collecting data is trying to anticipate all of the ways that bias can creep into your data, all the ways the data could become invalid because you just didn't think about some way that something you were doing wrong with the data collection. Or sometimes it's not even something you're doing wrong with the data collection, but it won't work for the question you want to ask. So sometimes the way you collect data works for one question and not for another. So one of the things I did was get some precision instruments. There is one datum that is very tricky to do and requires a lot of training, and that's actually figuring out whether a baby lizard is a boy or a girl. And there is a way to do that, but it requires a bit of a good eye for it. And so one of the ways I've ensured that those data are correct is that I'm the only person who does that. I do sometimes have a research assistant help me, but then I always double check the data. But we also write down who collects all the data so that we can go back and look at that through statistics to see if individuals are collecting data differently. So for example, when we measure the length of a lizard, even if you use a high precision calipers, sometimes individuals are just a little bit different when it comes to millimeters. And I can tell you, for example, that my lizards are longer than my research assistant's lizards. And I think that's just because I err on the side of overestimating their length. So sometimes it's impossible to completely control for things, and so we just try to keep track of all the potential biases and then control for it later when we do our data analysis. Yeah, there are things as well, like because we're always waiting for eggs to come out of the females, for example, we have to keep track of when a female has eggs inside of her, which is called gravid, when a female is gravid. Now, if she has a small clutch of eggs inside her, we might not notice that she's gravid because it's not as obvious, which means that we could miss the smallest clutches and only catch the biggest clutches. And that would be a bias. So we have to not only keep track of when the females are gravid, but we have to check all the cages every day to see if anybody has laid eggs in case we miss a clutch. You can just feel them. Yeah, feels like they have little jelly beans inside them. I think one of the funnest parts about being a scientist is when you finally have all your data together, and now it's time to look at it and start getting answers to your questions. So yeah, the first thing you do is look for problems with your data, and that might be as simple as a typo when you were typing data in. So I always do initial visualizations of the data, regardless of how many data points I have. I'll do scatter plots, I'll do bar plots, just to get a sense of, are there some data points that are just off by an order of magnitude? Did someone miss a zero somewhere when they were typing something in? And then I'll go back and look at the original data sheet if I think it's been entered incorrectly. But also getting a sense of the distribution of the data. Is it normally distributed? Is it skewed? How are things related to each other? So say I've measured mass and length in lizards. They're usually correlated, so I'll just check to see, okay, yeah, those two things really are correlated, so anytime I do statistics I have to keep that in mind. And then I will have, once I've done all that data checking, then I proceed on to my actual hypothesis testing statistics. There are times when you're just visualizing the data, so you might have a whole bunch of things you've measured, and you might just run correlations through them all and have a look at it, and then you're like, wow, those two things are correlated. And it sort of raises all these questions, and then you think, well, if those are correlated, maybe it's for this reason, and well, I can see if that is true by checking this other thing in the data. And you can kind of go down a few rabbit holes looking for other things in your data as well. So it is, sometimes you do serendipitously find other things in your data. So I do some very complex statistical analyses, because now that we have computers to do all of our statistics for us, we can do very complicated things. But at the base of it, a lot of what I do is similar to a t-test. So let's say a lot of my experiments have been, I'll hold some parents at warm temperatures and I'll hold some parents at cold temperatures. And I want to know if so then they produce eggs, and I want to know if their offspring have different sex ratios, or if they're different mass, or if they grow differently once they hatch out. So I want to compare the hot parents to the cold parents in terms of their offspring traits. So comparing two groups like that is a t-test. So I do something akin to that, but I add in various other things. So it becomes a slightly more complicated statistical test after that. I've worked with both small data sets and large ones. Typically, the data I collect are in a small data sets, just because it's hard when you're doing experiments to get really big sample sizes. But I've been working with these lizards for 5 years now, so I've been accumulating a much larger data set. I've also been doing other projects, so we just compiled a really large data set that looks at all of the literature investigating how incubation temperature influences the traits of offspring reptiles. So every experiment that's ever been published where they took eggs, incubated them at different temperatures, and looked at what happened to the offspring, we've extracted all of that information from the scientific literature and put it into a big data set, and there's 10,000 rows of data. And so we've been working with that as well to understand on a more global scale what temperature means for reptiles. There are limitations working with big data sets. I think the first is you can never see all of the data. There's something very comforting about being able to see a data set that's maybe 50 rows of data. You can look through it, you can spot typos, you can spot errors. You're never going to do that for 10,000 rows of data. So you have to devise computational ways to find little errors in your data. You have to think about the structure of the data whenever you want to ask a question, because you're never really going to visualize a spreadsheet, so to speak. I do a variety of modeling. We do some data modeling in terms of predictive. So I've been collaborating with a few researchers studying animals that have sex ratios sensitive to climate. So there's a skink in Tasmania that has sex ratios sensitive to climate, and the bearded dragon as well. In Central Australia, the central bearded dragon has sex chromosomes, but at really hot temperatures, the individuals that are supposed to be males develop into females instead. So what we're doing with both of those species is trying to understand, under projected climate change, what the sex ratios are going to look like, to make predictions about that. So that's some data modeling I've done. I also actually do a fair amount of theoretical modeling, trying to understand the evolution of traits. We use sometimes data from animals to help inform the biology, but it's usually fairly theoretical. So with the bearded dragon, we have evidence from some parts of Queensland that there have been sex-reversed animals. And the group I'm working with now has been looking across a wider range of Australia to see where else sex-reversed animals are. And we're interested to know if the incidence of sex reversal is associated with recent climate change. And then as we project in the future, using the climate scientists' predictions for what the climate will look like, how much more should we expect to see sex reversal? Where will it be most common across the range of the species? Does it mean they're going to lose their sex chromosomes? How much sex reversal can you take before you just lose a sex chromosome completely? So you can imagine humans just losing their Y chromosome. And then everybody being XX, and whether you're a male or a female depending on something else entirely different. It's a very strange and unusual system. They don't switch back, so whatever temperature they develop at, they're stuck in that one for the rest of their lives. And it looks like these individuals that have male chromosomes but develop into females might have different behaviors as well. So it appears they're more aggressive and they lay more eggs. And part of the research that we're doing in this big collaboration is to understand what else they do differently. Do they have bigger home ranges? Do they migrate further? Do they have different thermal physiology? I mostly communicate my research to other scientists. So that is at conferences. I go to conferences and give talks, and then I publish papers in the scientific literature. I also communicate my science to the public through things like blogs associated with my papers, through my website where I talk about my research, and through my courses, through lectures. You definitely have to change the language depending on your audience. So I have not used the phrase temperature-dependent sex determination at all because that's a scientific term that we use that most people find difficult to wrap their brain around. But in science we have a lot of jargon. So you have to you have to think about how you phrase things depending on the audience, particularly when you're talking about things like male or female, which when you're shortening that is just sex. When you write a paper, it has to be very technical and it has to show a really broad and deep understanding of the literature and the concepts. So it has to be quite sophisticated in that regard because it goes to other scientists before it gets published. And if other scientists think that you aren't representing the field correctly, then they won't let you publish it. So it has to be an honest representation of our current understanding of that field. So yeah, it has to be a bit more technical than what you have time for really with undergraduates. Undergraduates, I think, are capable of reading a scientific paper, and they do, but a lot of times you just don't have the time to get into the details. So you write a paper, and then you submit it to a journal, and it will go out to 2 other scientists that work in your field, at least 2 other scientists, and they'll review it for the validity of the science you did, so checking your methods to make sure you did it correctly, that they can't spot any obvious biases or ways that made your data invalid. And then they'll also assess whether you have interpreted the results correctly, so that you haven't said you've proven something that your data don't actually show. And then they'll also assess all of the writing and, as I mentioned, your coverage of the field and just your presentation overall. And they provide very detailed comments about what they think needs to be changed. There's always things that need to be changed. And then once the peer reviewers are satisfied that it is both technically sound, scientifically sound, as well as a good representation of the field and a good contribution to the field, then it'll get accepted and published in a journal. I love giving talks. I like short talks and I like long talks. I just love sharing my science. And it's very different how you give a short talk versus a long talk. And the questions that you get at the end, I find are usually questions where people have shown their interest. So I often find that I get questions where people just really want to know more. So I rarely feel like I'm getting grilled. Occasionally I don't know the answer, and I will say, as will most scientists, I don't know the answer to that. And if you're lucky, someone in the audience will ask you a question that you've never thought of before, and you'll realize, there's some other science I can do. So you often get really good ideas of where to go from questions in the audience. It's a really fun interaction, I think. It's to get feedback from other scientists, and everybody always thinks of different ideas. They see one piece of information or one bit of data, and people will take it in different directions. Their brain will connect it to something else they're working on, and they'll think, oh, well, what does it mean for this? And you don't work on that other thing, so you've never thought of that at all. It can lead to some really interesting collaborations as well. So not only ideas for your data set, but ideas for new projects. My advice for students thinking about pursuing a career in science would be to find what interests them the most, what they have the greatest passion for, and what they just can't stop thinking about, and to run with that and get as much training and interaction and mentoring and experience they can in that line, and just keep going for it.